Good evening to all of you who are present here and to Moshri ma'am also. Though we have not actually starting the meeting officially, but I already can see there are a lot of participants who are joined. So this is I just thought of informally greet you once. So good evening once again to all. And I think ma'am, Dr. Moshri madam, are you online? So I think ma'am is not joined yet. Okay, Mosri ma'am, are you there? So I think we can start now. It's around 6 p.m. Okay, so a very good evening to all of you, all the participants and resource persons who are present here today in the sixth day of the Atal online FTP program. So with the divine blessings again of Lord Swaminarayan Bhagwan, Param Pujya Brahma Sarup Sri Sri Hari Prasad Swamiji Maharaj, Pragat Guru Sri Sri Prem Swarup Swamiji Maharaj, and our beloved mentor and president of Atmiya University, Param Pujya Sri Sri Tyag Vallabh Swamiji Maharaj. We welcome you once again to this 6-day online FTP program. Today we have reached the 6th day of the online event. So as per the traditions, and as we are all doing it, so today also we will start this session with Omkar. So I request and invite all of you to kindly join me in the chanting of Omkars. Om. 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 Thank you. So I wish you all a very happy Makar Sankranti. Wish this festival brings new dimensions of prosperity, success, happiness, peace, and love in all your lives. So wishing again all a very, very happy Makar Sankranti, which is a very important festival for all of us. Okay, so we can now begin the technical sessions. I like to inform all the participants here that there is a slight change in schedule today because of some kind of logistical and time issues. There is a switch or swap among the resource persons today. So, Dr. Swati will actually start this session, followed by Dr. Abhijit. Okay, so before Dr. Swati actually begins her presentation, I just like to introduce her with a brief profile. So Dr. Swati is a seasoned biopharmaceutical professional with over 20 years of experience in drug discovery and development within the immunotherapy and biologics domain, primarily focusing on oncology and autoimmune diseases. Her expertise includes steering biological discovery in early phase development, assay developments, and validations, CMC, drug quality assessments, and drug regulations. Dr. Patel's expertise spans strategic management of bench to bedside transitions, understanding of clinical and translational research, and meticulous oversight of biologics life cycle, ensuring quality compliance aligned to stringent regulatory guidelines. She bears a strong foundation as PhD in immunology and oncology from ACTREC Tata Memorial Cancer Center, India, and she has completed her master's in analytical and medical chemistry, University of Mumbai. She has led biotech innovations encompassing monoclonal antibodies, liposome-based therapies, and vaccine development. Currently, she has actually moved to Queensland, Australia, and presenting here today as founder and principal consultant of BioEssence Consultancy Services and working alongside with University of Queensland as a research manager, managing the development of antigen-specific immune tolerance inducing, in short, ASITI therapy for autoimmune diseases. Her topic today is introducing consultancy services for your biopharma business success. With this introduction about her, I welcome Dr. Swati to kindly present her talk today. पार्थ भाई आप डॉक्टर स्वाति को प्रेजेंटर राइट्स दे दीजिए. Dr. Swati, you are the presenter now. You can present your slides.
Yep. Good evening, everyone. And I'm very happy to be here presenting at the FDP session today. And for that, I would like to thank Depashis for inviting me and Atmiya University for giving me this opportunity to be here. Let me just share my presentation here and then we begin with the presentation. And before we start, happy Makar Sankranti to everyone.
Wish you the same. Yeah.
I hope you can see my slides. I will put it in the slideshow. Yep.
Yes, it is. It is.
Yep. So thank you once again. And my talk is slightly different than what you all are hearing from last 5 days. And I have been present in some of the talks in especially the first session and some part of the first sessions. Today, I will be talking more about my BioEssence consultancy services and about my expertise and how we can assist the laboratories or companies in developing their therapeutic molecules. So, okay, yep. So before going into detail, so BioSense Consultancy Services is a biotechnology consulting firm. This is to support and accelerate the discovery and development of innovative biotechnology and biological therapeutics. Our team has extensive experience and expertise in developing diverse biological drugs, vaccines, and innovative drug delivery systems. Innovative drug delivery systems. We excel in the cutting-edge therapeutics, including immunotherapy, Diagnostic, which is basically the molecules combined with the radioactive molecule, and that can be used as a diagnostic as well as the therapeutics. And the cell therapy, gene therapy, and the associated challenges in the drug development pathways. So the vision of the BioSense Consultancy Services is to accelerate the transformation of potential innovations for the patients across the globe through collaboration and commitment. Our commitment in the high quality services, customized solutions, and adherence to regulatory expectations. Now, who can be the customers for BioSense Consultancy? It can be any biotech company or research organization, those who are working on the research and development of therapeutic molecules for the treatment of any kind of disease. It could be university labs or academic institute who are working on the discovery aspect for the treatment of the different diseases, and they would like to go further into development of their product or that therapeutic molecule further in the regulatory aspects of the drug development pathway. It could be any startup or spin-offs who would like to set up that R&D lab or quality control lab or manufacturing facility or quality management system, or they need some regulatory guidance to move forward in their startup or the spin-off. Okay, so before going into any details, it is very important to understand the consultant's profile and professional experiences before anyone can consult or get contacted to them, get in contact with them. So I will not talk too much about it, but I would like to present my expertise in the last 15 to 20 years. So I have done my bachelor's in microbiology and biotechnology, completed in 1996, and master's in analytical and medicinal chemistry. After completing master's, I was teaching for about 3 years, and I really liked teaching during that time period. However, my dream was always to join the pharmaceutical company, and that's where I first moved to the pharma base India and started working as a formulation analytical chemist. At that place, I learned that I need to be PhD, and the PhD is something which I should pursue to progress in the pharmaceutical company. And that's where I joined Cancer Research Institute, which is now known as the ACTRECT in Tata Memorial Center, as a senior research fellow funded by CSIR India. I worked there for around 9 months on the project, which was involving the screening of anti-cancer drugs. I then moved into PhD in immunology lab, focusing on immunology and oncology aspects of the non-small cell lung carcinoma and understanding immune regulations in these patients, and if we can use the COX inhibitors to reverse the immune suppression in these patients. I completed my PhD in 2008, and then I worked as a senior research fellow in the same lab, which was funded by DBT. During that time period, I worked on the development of therapeutic vaccine for cervical cancer, which was derived from the capsid molecule of the human papilloma virus. It was a multicentric project governed by DBT and funded by DBT. Soon after that, I started my journey in industry. My first professional experience was in Biocon Bangalore as a principal scientist. I worked in Biocon from 2008 to 2013, and where I learned most about the drug discovery and development part, about different type of asset developments, and I contributed in development of anti-CD6 monoclonal antibody, which is currently marketed as Alzumab in India. And I also worked along with that, many other molecules, which are fusion molecules, a different type of antibodies, and also the peptide kind of molecules. I then moved to biologics, to Acutest Biologics as a department head in Mumbai, and then Agile, then Apotex Research. And then in 2017, I moved to Australia. In late 2017, I moved to Australia, and I joined University of Queensland and Scientific Officer Immunotherapy. Here, my profile was to manage the ongoing research on the antigen-specific immune tolerance-inducing liposomes. So I will be talking about this in more detail when we move forward in the presentation. I also have experience as a lead quality control scientist, where I worked with the Thermo Fisher Scientific in Brisbane for around 1.5 years, managing the different quality control activities for the manufacturing purpose. I've worked with the Vexas, which is another cutting edge technology producing HD map, which is high density microarray projection map projections, and which are basically patch, which is similar to Velcro patches, but in the metal form, and then that can be used for loading the vaccine on those microarray patches, and then deliver those vaccines self-administered by people on their own subcutaneously. So this is something to make the vaccination process easier and self-administered. So my role here was leading a team for the development of formulation. We worked on developing the existing formulation, making it more suitable or optimized to load them on the microarray patches. And then again, I came back to the University of Queensland and get involved in the development of antigen-specific immune tolerance therapy. In 2023, so I'm currently working here at the University of Queensland, and along with that, in 2023, I've started my own consultancy services, which will assist in transforming potential drug candidate into developing into the therapeutic molecule. I'm not working alone. I do have my team members, but at the moment we are all working from the different places. I have my colleague working from Germany. I have two of my colleagues working from India and one from USA. So we work together, and we have been working from around 1 year as of now, as in consultancy services. So yes, so going into detail, what kind of services we provide. So as you can see here, we can assist you in the development of all different aspects of the therapeutic development, which can start from the discovery, drug discovery, R&D, QC lab setup, quality management system, and as we move forward, the manufacturing, toxicology, regulatory affairs, clinical trial stages, and biomarker assessment, identifying responders, non-responders for your trial, and also the project management. Most importantly, the asset development, qualification, validation as well. I will be talking about this drug discovery pathway in more detail for those people to understand who are not directly involved in this process, so that they can understand the different steps of the drug development processes. So here is the drug discovery and development process at very high level. The actual process is much more complex than what we can see here. I've tried to simplify just to understand what happens in the different stages and how it progresses from a lab-based research to the market available drug. So it all starts with the discovery. Someone identifies that if we use a particular molecule, it binds to a so-and-so molecule on the cell, within the cell, or the free moving molecules in the cell, like free cells, and it gives a certain outcome. That particular outcome can be used as a treatment for certain disease, and that becomes mode of action. So it's completely knowledge-based target validation, that you understand certain process, how the particular cells or disease progresses, how we can use a particular molecule to treat that disease. And for example, you find you have a compound which can bind to a receptor, and then it can lead to the proliferation, or it can lead to the apoptosis, and then that can be used for the treatment of cancer, or that can be used for the treatment of certain other type of disease. And then it goes into the different aspects of developing that idea into a complete treatment. And then the journey of drug development starts. So you understand the mode of action, you understand the molecule which you're working on, the drug which is you're working on, and then you identify your target, where it binds, how it binds, and how it gives a particular effect. You identify biomarker, you have an experiment or assay to identify a certain biomarker. You can do the in vitro efficacy assays, that is in the cellular 96-well plate or in the Petri plate. You try to understand whether it shows any effect. For example, if you're working on the cancer cells, you can incubate your compound with the cancer cells in the 96-well plate, and you see if it inhibits the growth of those cells. So that becomes your in vitro efficacy assay. You try to understand the potency of the drug, like what is the dose it is effective, what is the strength of your particular dose, and how much effect it can lead. So these kind of like basic understanding gives covers under discovery. It then moves into CMC, that is chemistry, manufacturing, and control. So once you are sure that yes, this is how it works and this is what it does, and it can be used for the treatment of so and so disease, and then it goes into understanding your product, like what are you looking for, what is the pH it works for, what is the structure of the molecule, how it can be manufactured, how it can be manufactured from lab to the big scale where you want to produce for the entire population. What should be the concentration, how you can identify that your drug is pure, how you can identify the impurities present in the drug. how you can identify the strength, the safety, whether it is contaminated, it is not contaminated, whether its endotoxin is present or not, and the formulation studies, which buffer is the best, how we can improve the stability, how can it be stable for the longer time. So all these studies are covered under CMC, including the manufacturing aspects. It then moves into the non-clinical phase. Now, at this phase, we try to understand the maximum pharmacokinetics and pharmacodynamic aspects in animal, and especially the safety aspects of the drug. So it involves the in vivo efficacy model. You have the animal models where you try to understand and give the different doses, and you try to understand whether the drug is effective or not. You try to get the acute safety profile, ADME outcome, like when it gets absorbed, distributed, metabolized, and excreted from the body. PK and TK studies, like how the concentration of drug increases in the body. The repeat dose safety study, where when you give the repeated dose, whether it's toxic or not. Dose range finding, which basically involves the finding of minimum effective dose, that what should be the minimum dose which you should be using for the treatment, minimum toxic dose, and the maximum safe dose, which are identified by the toxicology studies. At the same time, when you are working on the CMC and non-clinical phase, you also need to understand and establish the quality of your product, like what is the target profile of your molecule, what should be the pH, what should be the concentration, how do you identify that. All those quality control aspects needs to be confirmed during this time period, and that involves all QC experiments, efficacy assays, stability assays, and CMC assays. It then can move, this can also happen along with the phase 1 trial, provided you have fairly good data to move forward in the phase 1 trial. So phase 1 trial is basically first in human trial. It more focuses on understanding the safety of the drug than the effect of the drug. It is done in the smaller number of subjects, like around 10 to 30 subjects may be sufficient. And the selection of subjects based on the mechanism of action, identification of biomarkers, safety assessment, and PK/PD data is generally the primary endpoint of this phase 1 study. And then once we have the confirmed safety confirmed, and during this phase, and we have some efficacy outcome, we can go into the clinical phase 2 and then clinical phase 3. So basically, clinical phase 2 and 3 differs mainly in the number of subjects, number of patients getting involved in the study. Phase 2 will have around 100 to 200 patients and subjects, and phase 3 will have up to 1,000 or 2,000 subjects. So it basically involves the all different aspects and aspects which are related to the drug mechanism in the body, selection of subject, monitoring of biomarker, safety assessment, efficacy analysis, risk assessment, all these aspects. If the drug is found effective, sufficiently effective, and safe, then it can move into the clinical phase 4, which is called pharmacovigilance monitoring, and at the same time it can be launched in the market. So the pharmacovigilance happens in the market scenario, where the doctors will have the one-to-one assessment with the patients, and they will note down any side effects or any long-term effects will be noted down. So that goes on for many years, and that is specifically to understand the long-term severe adverse effect identification and the risk assessment for that particular drug molecule. So this is basically the entire drug discovery development process. There are a lot more things associated with this, especially the regulatory guidances. All these steps require to be done as per the regulatory guidance, as per the regulatory requirement, need to have particular system setup like GLP laboratory, good laboratory practices laboratory, GMP laboratory for the manufacturing purpose. You need to have all different type of documentations, and all the activities needs to be documented as per the requirement. So it requires a lot of efforts and a lot of time, and of course money. I have put a time period and money here, which is a very vague number, which is not, it all depends upon the type of the drug and the type of the molecule you are dealing with. But this is a rough timeline we can look for. So it can fairly take around 10 to 15 years for one drug to be in the market after the discovery is made. So the one thing is Biosense consultancy services can assist and can guide you through each of these steps up to clinical phase 2 or some aspects of clinical phase 3 for especially the bioanalysis and related to assay development and validated assays and those kind of things. We can definitely assist in the early development part and discovery part and all different aspects of this two part. So the next slides will go more into the details of this requirements and services we can provide based on our expertise. I will not go into too much detail of this, but I'll just cover the high level. All details are also present in my website. You can always have a look at the website and get in contact with me if you need to. So basically, we can help in establishing labs, R&D labs, or the quality control laboratory. The R&D laboratory and the quality control laboratory requires a different quality management system and different regulations to be established. And our team can help you guide through the requirements and how to implement those requirements. We then have the quality management system. Even though you have the specific facility already set, there are the good quality management systems need to be in place, which confirms all the proper documentation, review process, approval process, any change in the process need to be identified and recorded, and how to manage the entire quality management aspect is what can be covered. We can assist you in gap analysis, risk assessment, audit readiness. So the regulatory authorities do perform the audit of the manufacturing facility or the laboratories time to time to make sure that the functions are going as per these standards. And our team can help you write SOPs, create forms, test methods for the quality control test, then qualification, validations, protocols, reports, and batch records, sampling plans, specifications, and different operation processes. Sorry. We of course can help you in drug discovery studies. So once one have identified that a therapeutic molecule works in a certain ways, you need to perform the efficient way of experimentation where it can give you more conclusive results and more confirmed confident outcome, and that makes you the process faster and easier. We can definitely help in molecule selection, in vitro, in vivo assays, planning experimental designs, and that mechanism of action, and all other different aspects of the discovery as well. Strategic planning for the drug development, as I said, it requires a lot of understanding of the regulatory requirement and the formulation development, stability development processes, technology transfer for manufacturing, different contract and the outsourcing activities. So all those can be outsourced to consultancy services like Biosense. We can definitely assist in assay development, qualification, and validation path for QC assays or efficacy assays, or the simple drug discovery assays. Project management, it is very important to manage these projects efficiently so that although it takes like 10 to 15 years for the complete development, if we do not plan it properly, it might take much longer than that, and then someone else might have the similar discovery and they launch their drug in the market. So it's very efficient. You need to be very efficient and plan it in a way that you can be ahead of the market and produce your drug. These are the different type of assays which I personally have experience working with, but I will not go in the details. It basically covers all different assays for the monoclonal antibodies, peptides, biological molecules, Diagnostic, some formulation assays for the thermal stabilities for the different vaccine, some specific assays for the antigen-specific immune tolerance-inducing liposomes. I will be discussing about this, some of these data from this. And how biomarker identification can help into good outcome of clinical trial. So this is my PhD work. This is to highlight my experience in terms of understanding immunological aspects and different type of experimentations and the way we do the experiments in hand. So I have, my mentor was Professor Shubda Chiplunkar, who is now retired. My project was to understand IL-10 and PGA2 mediated immune dysfunctions in non-small lung carcinoma patients. So I used to collect blood samples from these patients and then separate PBMCs, incubate PBMCs with the different T cell, B cell activations and like CD3 and other molecules, and understand the cytokine balance, proliferation, cytotoxicity outcome, reactive oxygen species generation. So basically understanding all the different aspect of this ERKP38 MAP kinase pathway, which is also regulated by COX2, and the COX2 production, COX2, which is a cyclooxygenase 2 enzyme, leads to the production of the prostaglandin E2, which is then acts back on these cells and leads to the overexpression of this pathway. This in turn inhibits the immune responses in lung cancer patients and allows tumors to grow. And then this is how the tumor makes imbalance in these patients and sustains for the longer time and grows for the longer time. So if we try to inhibit COX2, will it revert the cycle, and then can we remove the tumor from the body? So that is what my project was. And I used a battery of around 9 to 10 COX inhibitors and incubated and conducted the same experiment after COX treatment, and we see a significant reduction in the PG production and then also the impact on the other cytokine outcome. So basically, I elucidated the complete signaling cascade for induction of immune suppression by tumor-secreted PGE2 and induction of anti-tumor immunity by COX inhibitors in non-small cell lung cancer patients. Of course, using the ex vivo cells and not the in-human trial. So that concluded that the COX inhibitors can be used for the management of the lung cancer by balancing pros and cons of the COX inhibitors. So COX inhibitors, those who are not aware, are basically all those painkillers which we take: aspirin, paracetamol, indomethacin, celecoxib, rofecoxib. All these are COX inhibitors of the different strength, and they do have slight different functions in the way it reduces pain. After my PhD, as I said, I worked briefly for around a year for the development of therapeutic HPV vaccine. At Biocon, my major project was developing monoclonal antibody, which is itolizumab, which is an anti-CD6 antibody. It targets the CD6 molecule, which is a classic co-stimulatory molecule on T cells. And it is currently marketed as an alzumab for the treatment of psoriasis. And I was involved in the complete development from the discovery until it went into the market, got approval to be launched in the Indian market. I was also involved in the clinical trial, sample analysis, some of the potency assay development, and all different aspects of the development. And the studies are published in the public domain and as the publications, and you can always go back and have a look at details. So one of the aspects here was to develop the biomarker assay for the clinical studies, where we wanted to understand whether the psoriasis patients are having any effect of this anti-CD6 monoclonal antibody. So what we did is so as you know, psoriasis is a skin disease, an autoimmune disease, where the patient's immune system acts against keratinocytes of the patients. So keratinocytes are basically the below layer of the skin, and because of that, the skin gets thick and it also becomes itchy. There are also many other symptoms associated with that, but these are the main symptoms. When you see this, you can identify that it is psoriasis. So we collected skin biopsies from the patients when they visited first, and then started treatment. And at the end of the treatment, we again collected the skin biopsy, and we looked at the different thickness in the skin. So this is where we could see that the thickness of the skin significantly reduces from day 1 to day 85. We also looked at the number of CD3 T cells. So basically the immune responses are through CD3, CD4 cells. So we see a significant decrease in the number of these cells in the responders. However, there were some non-responders who did not show any response. And this is where the drug development aspect comes in the picture again. One need to understand that why these patients are not responding and what we can do so that these patients can also get benefited by the treatment. So that even though you have a good outcome, that can become your next research project. This is one of the assay which was developed at Accutest Biologics for the measurement of human growth hormone in the healthy individual serum. I will not go in the detail. This was mainly of the assay development and validation of the bioanalysis method for the biosimilar, which is a human growth hormone. So human growth hormone is used for the treatment of Turner syndromes and in those children who have some growth deficiency. And it's already in the market. Those who want to produce the biosimilar molecule can need to take the BAB study, where they can give they can give the biosimilar molecule to the healthy individual and then look at the pharmacokinetic and pharmacodynamic outcome, which needs to match with the drug which is already in the market. So this study is also published and it's in the public domain. I have included all the studies here which are already published and in the public domain. Because of the confidentiality issues, I cannot present anything which is not published. This is the main and most challenging thing which I feel is important, which is detection of antigen-specific T cell responses. So currently, this is the project which I'm working with the University of Queensland. It's a development of therapeutic molecule which is called ACT, which is antigen-specific immune tolerance-inducing liposomes. So this is basically used for the treatment of autoimmune disease. So in autoimmune disease, if you're aware that body's own immune system reacts against some of the part of the body's own part of the body, like molecule or cells of the body, and that gives the severe symptoms. And then it's a constant, because the antigen is part of your body and your own immune system is acting against it. So it is something which always goes on, and there are only immunosuppressive treatments are there at the moment, but there's no root cause. Like there's no way we can reverse that immunity. Like we can stop immune cells identifying that as an immune antigenic response. So this is what we are trying to do here. So we are trying to teach T cells to not identify that as the antigen. So currently our studies are progressed very well with the two disease, type 1 diabetes and rheumatoid arthritis. So how this molecule works, it's a liposome molecule which contains two active components. One is peptide from the patient. Now this peptide is the one against which the immune cell is recognizing is producing an immune response. So basically, if it is type 1 diabetes, it's an insulin against which the patients are generating antibodies, and the insulin producing cells are getting killed by the own immune system. So that becomes an antigenic, insulin becomes an antigenic peptide. So we use insulin, or the part of the insulin, as an active component of this drug. And then we use another molecule, which is NF-kappa B inhibitor. So NF-kappa B pathway is a very important pathway in the cell activation in immune cells. So now what happens is that the peptide component is encapsulated in this center part, which is an aqueous core, and the NF-kappa B inhibitor, which is a lipid bound molecule, which remains in the outer layer of the lipid in the liposome. When these liposomes are injected in patient, the liposomes are taken up by the antigen presenting cells. They migrate to the draining lymph node, and within the draining lymph node, the antigen presenting cells present the liposome peptide, which is there in the center, to the antigen specific T cells, and simultaneously the NF-kappa B inhibitor try to suppress the T cell activation. So what happens that specific antigen specific T cells now becomes non-reactive to that particular antigen. So then when this happens, if impact get translated to the other cells, immune cells as well, it goes into the regulatory cells, the regulatory cell numbers increases, and then you will see decrease in these symptoms of the autoimmune disease. So this dual mechanism results in the inhibition and regulation of the peptide-specific T cells, promotes tolerance-inducing immune responses, and this process leads to reduced inflammation and decrease in autoimmune system. So now what we wanted to do here is type 1 diabetes is a disease which we identify. So one of the indicator of the diabetes is the glucose level. So when you see that patient have a high level of glucose in their blood, there is a diabetes. So there is either the insulin production is low, it could be type 2 diabetes, or it could be type 1 diabetes, where the insulin producing cells are getting killed by the immune system, and then there is no production of insulin, and that leads to the high concentration, high level of glucose in the blood. But by the time this can be identified by measuring the blood glucose levels, it's too late, and almost 90% of these cells, insulin producing cells, are killed by the system. So if we want to use this treatment, we need to use it very early on. So one of the challenge is to, like, when should we treat patient? When they come in the clinic first time, when the glucose level is high, or we should try to get those patients who have hereditary of type 1 diabetes, who have issues with the related to those, and who have that particular genetic composition, are likely to get the type 1 diabetes. And even after they identified for the diabetes, when should we treat? Can we treat immediately? Should we treat little later? So one of the aspect was the biomarker, to develop a biomarker assay for the phase 1 clinical trial in the selection of potential responders and efficacy indications. So basically that is called responders, like who will be benefited from our clinical trial. So the critical issues here were the identification of responders for such highly antigenic specific immune response. And also what happens in the early development is, like, it's a very new drug. We are not aware what will be the dosage will work in the patient, and it might happen that we do trial with the low dose, and then we do not see any indication in the glucose level, but we will not understand that we are not seeing the effect because it is not working, or because it is working but it's not sufficient. So we need to understand the immune markers here, that what are the biomarkers which can indicate that yes, the treatment is working, but we might see the response later, or we might need to increase the dose, or we might need to optimize it in a different way. So those are the challenges. So identification of efficacy during the trial Identification of efficacy during the trial as immune modulatory signal can be missed as the therapeutic strategy. That could be because of the dosing, administration, timing of dosing, which is not optimized when you do the first trial. Having an assay to measure the antigen-specific T and B cell responses can help overcome these challenges. The lack of sensitive and reproducible methods to analyze antigen-specific T cell responses is generally a challenge in this kind of things. So we try to work on the proliferation assay using the CFSC dye to detect responding T cells in these patients and to understand like if there are more responding T cells in some patients, then there are more possibility that those patients will be responders and will have good outcome from this treatment. So this CFSC assay is a very traditionally used assay. And what we did exactly and how it works is like we took blood from the patient and isolated PBMCs, T cells. We label these PBMCs with the CFSC dye. So CFSC dye is one, it gets into the cell and it remains in the cell if the cells are alive. When these cells divide, the next generation of the cells will have half the concentration of the actual labeled cells, and because of that the intensity of the CFSC dye, which fluorescence under flow cytometer, will reduce to half. The same thing will happen to the next cycle of division, so it again goes half. So every division the intensity of the CFSC dye will reduce to half, and this helps us identifying whether the cells are proliferating, whether the cells are dividing in response to the peptide which you have used, or they are not dividing. If they are dividing more, that means they are more responsive, and if they are not dividing, that means they are not responsive. So this is the basic assay which we thought we can use because we were interested in specific antigen specific T cells, antigen specific CD4 T cells, and that is a bit challenging when we want to do it. So flow cytometry was the best method where we can also in future identify whether it's a regulatory cell, whether it's an effector cell, whether it's naive cell, and what is the subpopulation of that particular responding cells. So we conducted an initial experiment with the different peptides. There are different parts of the insulin, proinsulin, different sequences, and we incubated patient PBMCs which are labeled with the CFSE, and we saw the responses. So this is cell division index. I have given the formulation in the next slide, but basically what it means that higher the cell division index, the more number of proliferation cycle has happened in that particular peptide wells. So as you can see here, there is one well. This one is unstimulated cells. These are all different peptides. And this one is with CD3, which is a very strong stimulation for CD4 cells. So as you can see that this particular peptide have responded well. So we selected that peptide and then we conducted study in the patients. So here I have, this basically explains how we did the gating strategy and how we calculated the CDI value. We also need to decide the cutoff of the CDI value, like a cell division index value, because even if we do not stimulate these cells, because we are taking them from patients, they may get exposed to the many different antigen in the environment, and they do have some basic level of cellular division happening even without any stimulation. So that needs to be removed. So we remove that non-specific part and then we worked on those aspects. Those who are interested can read this paper and go through it. So here you can see that after doing that optimization, you have no peptide wells where the CD4 positive cells cell division index is very low. This is one is PI33 peptide in response to PI3363 peptide, and this is in response to CD3. So you can clearly see the response, and then there is an initial gating first, second, and third. So these are the optimization strategy, and you can see when we revise the gating strategy, we could better identify and observe the stimulation with this peptide. So then we started doing screening of the patient using this technique. So we also performed all these different type of optimization. It took a long time, and we published the whole paper for the optimization of the assay. And this is where we used the assay. We took 32 type 1 diabetes patients who were just recently identified having diabetes, less than 3 months. 40 patients were we knew more than 3 months having type 1 diabetes. 17 pre-diabetic antibody positive cells, those the hereditary kids or the sisters or brothers who who do not have diabetes, but they are likely to have diabetes because they already show antibody in their blood. Then there are 9 antibody negative first degree relatives, again like kids or sisters or brothers who do not have diabetes and they are not having any antibody in their blood, but their parents or their siblings do have diabetes. And 16 healthy controls. So as you can see here, when we started measuring the cell division index, you can see healthy control, that cell division is like within 1 to 10 cycle around like 5. With antibody negative, it increases slightly because they do have that immune responses, although they are antibody negative. And antibody positive have even more response. These are type 1 diabetes patients which are diagnosed within 3 months. And these are the one which have already crossed 3 months. So as you can see, the best effective it could be is the one which is recently diagnosed within 3 months. So if we are conducting a trial for type 1 diabetes using this therapy, we should use these patients which are just recently diagnosed within 3 months. So this will help us moving forward in the trial. Like suppose if we conduct trial with type 1 diabetes patients who have diabetes more than 3 months, then we likely not see any response from the patients and we think that the drug is not effective. But it is not like that. It might be effective to the patients who are recently diagnosed, but not to the late diagnosed. So this is what we need to identify for these kind of specific therapy. Who are the responders, who are not responders, and this will be very useful going forward into drug development. So this is a similar kind of the study when we used rheumatoid arthritis patient. Now the rheumatoid arthritis we used we monitor the immune antigen specific T cells in the whole blood. I don't know how much time I have left. I have half an hour.
Dr. Swati, you have still 10-15 minutes of time. So
okay, great.
Yes,
so I
ma'am.
can
Yes.
go into some detail.
Yes,
yeah,
yes, ma'am.
not a problem.
Yes.
Yes, sure. So, Eva, so this is a similar thing we did for the rheumatoid arthritis. Now, rheumatoid arthritis is a different disease, and we need to have a different strategy for this. So, one of what we found best for these patients is the evaluation of the fit for purpose assay, and that is to monitor antigen specific functional CD4 T cells subpopulation in blood using flow cytometry based assay, which is specific to the peptide MHC class 2 tetramer. I have missed putting a tetramer diagram here, but basically what the tetramers are is basically a molecule which has 4 different arms, and it can bind to the 4 different receptors. So the tetramer will have the specific antigen bound to it to the 4 different arms, and then that can bind to the T cells and the 4 different. So what happens is that like 4 different T cells can bind to the tetramer, and tetramers are labeled with the streptavidin and biotin, so they can be identified with the signal from streptavidin, biotin, and related fluorochrome. So that will basically help identifying antigen specific T cells. So when we are developing novel antigen-specific immune tolerizing therapies that target antigen-specific CD4 T cells for the treatment of autoimmune disease can be benefited significantly by these kind of assays. Antigen presenting recognize antigen specific CD4 T helper cells recognize peptide major histocompatibility complex PMHC class 2 molecules of antigen presenting cells. They activate and sustain autoimmunity by cross-linking with APCs to activate CD8 positive cytotoxic. And we don't need to go through these details. So basically, we wanted to calculate the number of circulating antigen specific T cells that can serve as a response biomarker if we undergo the clinical trial. But it is very challenging because at any given time period, the antigen specific T cell numbers are very less. And they are very less and they have low frequency, which is in circulation. They are lower in sensitivity and specificity. So even though they are antigen specific, they can cross react with the other T cells. There is high cross reactivity between the T cells and they are low sensitive. And autoreactive T cell receptors typically have lower avidity in peripheral blood T cells. So basically, what is avidity is the strength of binding any binding to the receptors. So if the avidity is low, what happens is they tend to detach easily, and that is dictated by avidity. So if there is a low avidity, it can easily get separated, and that is very difficult if we have assay where they easily get separated. You get very variable results. You don't get proper results if you are using that. So those are the challenges, and we wanted to use these methods because flow cytometry is the one method which can allows you to identify different subpopulation in the circulating blood. So if we can do that, it's a very easy marker to understand whether it's working or not. So we developed an assay for enumerating antigen-specific T cells recognizing type 2 collagen epitope. Now this is, in case of diabetes, it was proinsulin peptide. In case of arthritis, there is type 2 collagen peptide epitope, which is presented by rheumatoid arthritis associated risk HLA-DR. And what we did is we evaluated we developed an assay and we optimized the assay using tetramers. We evaluated tetramers with the required precision and reproducibility, and different around 9 to 10 subpopulation of CD4 T cells were calculated and monitored using these tetramers. And we evaluated specificity, precision, and reproducibility for the research environment in non-regulated laboratories for 20 subsets of antigen-specific T cells from peripheral blood of rheumatoid arthritis patients. The assay worked excellent with overall precision of %CV less than 25% for intra-assay repeatability, inter-analyst precision, and inter-assay reproducibility. Some people might not be fully aware about these terminologies, but I think if there is a presentation happened with the assay development, the end qualification validation, these things will get covered into that. The precision of assay was good, and it was good for these kind of assays and for the initial research-based monitoring which we wanted to conduct. So now we are using this assay for the longitudinal analysis of treatment response and disease activity outcome in rheumatoid arthritis patients, and adaptable for translation and immunotherapy clinical trial settings. We have published this assay outcome as well, and you can look at the details online. So yes, we have the assay development and qualification data published as an evaluation of fit for purpose assay to monitor antigen specific functional CD4 T cells. We have conducted a phase 1 trial using these antigen specific immunotherapy, and we used this assay into this trial to look at the outcome. So even that paper is being published with the results of the randomized trial. These are the data which was we calculated. So I won't go into much of detail, but basically using these tetramers in phase 1 clinical trial, we could, so we used 3 different doses in phase 1 clinical trial. You can see here in figure B. We had placebo, we had 0.3 ml, 1 ml, and 3 ml dose. And we calculated different antigen specific CD4 T cells. And you can clearly see that and in placebo, the CD4 T cells are less. It increases with the with the time period. There is an increase in these cells, but then eventually it reduces. With the higher doses, it goes even lower. So these are the ways we can use these assays. So these biomarker assays are very important and play a very crucial role for, especially now when we have a very complicated treatments coming in market like cell therapy, gene therapy, these kind of ACT therapy or liposome based therapy, LNP based therapies. So for these kind of therapies, it's very important to have a different markers than the disease outcome, because it might take some while. Like for our therapy, we may not see results for immediately when the patients are treated, but we will see results after few days or after few months, because it takes a while that the whole immune system gets responsive to the treatment. So this is what I think it was important to see, that why it is important to have consultancy services or a guidance from the expert in this field. To, if you have something new coming up, if you are planning on the discovery, it's always good to get in touch with someone who is experienced in this field and who can guide you for planning different experiments, having the right experiments in place, having the right conditions for the experiments, and look at the right correct outcome of the experiment. Now, I've been doing this translation research management from last 5 years in Australia, and at University of Queensland, I've completed translational strategy, product development, management of translation research for type 1 diabetes and RA antigen-specific ACT liposomes. This included all different management, strategic planning for the trial as per regulation, guiding in the laboratory for doing experiment, product development, efficacy, formulation, all these different aspects. We completed this phase 1 trial in rheumatoid arthritis, the data which I have shown you in 2021. We started phase 1 trial in type 1 diabetes in 2024, late 2024, just 3 months before. And I am currently planning and handling the manufacturing process for the phase 1 trial for rheumatoid arthritis patients, which will start in 2027. And we have recently received a grant for this project for around $11 million to conduct this manufacturing and get completely ready to get into the trial. While I was working for the Thermo Fisher, I was monitoring and supervising the complete batch-related quality control aspects of manufacturing of biologics for phase 1 to phase 3 and pre-market batches. I completed 6 batches in 1 year. At Vexas, I was involved in the development of formulation for high-density microarray projection, which are called as HD maps, and based delivery system for existing vaccines delivered by so this is specifically for those vaccines which are generally delivered by syringe based mode, and we wanted to transfer that into HD maps. So yes, these are the publications which I have published throughout my career. I would rather say that I have some of the reviews as well, and being in the industry, we don't always get the chance to publish papers, but it's good to have some of the publications having lined up. This is again the services Biosense Consultancy can provide, and I'm very happy if any of your laboratories or your lab is currently working on any drug molecule, therapeutic molecules, you need guidance. It could be a small experiment plan or anything. I'll be happy to assist you. There is no project which is small for us. We can assist you in data management, data analysis, understanding the data, or planning experiment, or whatever is required to do to take that particular discovery further in the market. So with this, I thank you all for inviting me, especially Debashis for inviting me. Thank you all to attend this lecture and paying attention. These are my details. You can contact me, email me to my Biosense email. You can review my LinkedIn profile, my ResearchGate profile, or look at the Biosense Consultancy Service website to know more understanding of the services we provide. There is a small form. If you would like to contact, you can fill that form or directly contact me on my mobile as well. I'll be happy to talk to you. So with that, thank you. Thanks everyone. And I'm happy to take any questions.
Yes, ma'am. Thank you
Dear
so
Dr.
much.
Swati, ma'am, just, I will just finish it and then you can take over. Okay, so thank you, Dr. Swati, for a really very interesting and, thought-provoking session. Okay, so finally, my wish of, transferring your expertise to the audience has come true. So anyway, so you have given a very as I said, interesting presentation to the audience here, where you have started describing about your startup venture, okay, and the different kind of services you are actually providing to the different uh, customers in the biopharma pipeline. And you have very, I think the participants are really interested to see your journey, okay, from joining after your master's, okay, a biopharma or biotech industry, and your journey throughout where it's taken wings and now you are actually owning a bio venture company. You have also different touched upon or taken the audience through a very nice journey of drug discovery and development process, which includes from discovery to the CMC part, then the non-clinical phase, and ultimately to the different phases of the clinical research. You have shown the audience what is the potential for your startup, like the QMS quality management system, drug discovery studies, the strategic planning for drug development, and so on. You have also given a very nice overview of the different projects and the different, the research that you have been associated in your career, like the PhD work in the number of companies that you have actually been associated earlier, like Biocon.
You are muted, Debashish.
Okay, I'm really sorry for that. So as I was telling, in your present role, you have shown how the different types of research that you are involved in, like optimization of the method to detect an antigen-specific T-cell response in the type 1 diabetes, which is, I think, a very innovative study for the treatment of type 1 diabetes patients, where wherein you are working on a therapeutic molecule and quite an innovative one called ASITI, which is a liposome-based therapy. We have shown the optimization of a flow-based CFSE method to detect auto-antigen T-cell response, and so on. It's really interesting to show, and as you said in the last one, that you have published so many papers in very reputed journals, which is really rare, because as far as I am concerned, I have not seen anybody from the industry publishing so many papers, okay, which really points again to
Thank
your
you.
great depth and your research experience. And you have also shown about the biomarker, from you're working with the biomarker from the phase 1 clinical samples, and ultimately your translation research management. We are really indebted to you for this great, different aspects of the as such the biopharma uh, services and research. And once again, from the entire Atmiya University, we as coordinators and the FDP program, we are really grateful to you for taking your precious time out from your extremely busy schedule. I know, again, personally, how many of you are, how much busy you are. So thank you once again for coming here and delivering this talk. Part Bhai, let's have a good round of applause for Dr. Swati Madam.
Thank you, Devashish, for first of all inviting me here and giving me opportunity to present my work and to talk about my professional experience, my consultancy services, and I hope we work together.
Yeah, sure. We have been in the past and we'll surely do more in the future.
Ma'am, we have a couple of questions.
Okay, so Dr. Swati, now we are have some kind of questions now in the chat. Okay, with your permission, I just like to post it to you. Okay, so the first question comes from Dr. Ashish Kumar Singh, who says that you have looked for CD4 positive cell, but for which kind of inflammatory response? Is it Th1 or Th2?
Yep. So this is what when we are looking this. So in case of autoimmune disease, it's always like you have more of Th1 response happening in the patients, which is against a particular antigen. So we look for more of an antigenic response rather than Th1 or Th2. Like if we see in the beginning, if we see more of CD4 T cells in these patients, which are specific to that peptide. So that indirectly means that there is a stimulation of that CD4 cells to that peptide. So they are Th1 types. Mostly they are Th1 types, but there will be later on once we, so in the beginning there will, when we have more number of CD4 T cells which are antigen specific, there were more of Th1 types, which is like inflammatory types. But as we progress with the, so this is what we have seen in the mouse studies, when we progress with the treatment, with the more doses given to them, we eventually see more regulatory T cells coming up, which are antigen specific. And we also see that even though they are not antigen specific, the whole Th1 response get converted into Th2, or I would say not Th2, but less inflammatory response. So that's what happens. So we don't specifically look for Th1, Th2, we look for more of antigen specific outcome and the inflammatory markers, which is, of course, some of them are Th1, but we also look at the Th2, which will be help us identify whether there is a modulation of immune responses.
Thank you for your response. Now, moving on to the next one, Dr. A. Madhavrani asked that, ma'am, as of now, as a university level, we could do animal studies with rat or mice, but after we have no clue to continue further towards drug discovery. In what way can we proceed further? So she's just asking you about some insights into that. Can you please guide her in
So this is what, as part of the discovery lab, what we can do is max to the study in the rat or mice, or sometimes we have access to the rabbits and we can do that. And that's where like what happens is once we have the confident outcome, like we are sure that this is working, this therapy has some potential to get into the treatment pathway, we start doing experiments in mice and in lab in vitro, which more align with the clinical requirement. Like different doses, different timing of doses in mice, or different way of administration, and you find like which works best, and then you start summarizing those results, and then based on that result, we need to provide the non-clinical pharmacology report that will describe all these outcomes, and we go for the clinical trial. Now, before going for the clinical trial, we can do the toxicity study in animal, which is more to understand is there any toxic response in rat or dog, and we need to follow the regulatory guidelines. So this is when it follows. So that is why once you have identified something, it's very important even in the discovery lab that you do it very systematically. You fulfill the requirement of going into the next steps. And that's where you can get in touch with someone who is expert, who have experience in these kind of translation, and then move forward and do the experiments.
Dr. Swati, I just like to ask you one thing here, okay, as a thread to this questions. You know very well that in academic setup, we don't have all the, required infrastructure and, stuff there. So even if suppose, so it's actually very difficult practically if you think to, follow all the guidelines, regulatory guidelines and so. So, as Dr. Madhavani madam was asking, so would you suggest here to next for the next steps after the initial say the R&D part or the discovery part to tie up with any like company, even if it is a small startup or something, to follow the next steps, or it can be done in an academic setup also? What are your insights on that?
So you can definitely consult with someone like consultancy services. You can consult with someone like us who are helping in the translation research, or you can also collaborate with some academic lab who already have experience doing these kind of studies, who are also working in the similar field where you are working. So then you can go into the collaborative mode and then get some experiment done with them, publish papers together. So that's how the academic environment is. It's more of the publications and recognition of research, but that can be used for this kind of study as well. You do more collaborations and or you can also, if you have grant or money or fundings available, you can have consultant initially. You can get the guidance and then proceed for some time. It might be just small understanding or small troubleshooting kind of consultancy, and then move forward, have certain things done, and then it can be asked for the more funding. And so once you get into the drug development pathway, and that is why I have put those money requirement on the top, you will need a lot of money. That means even if you are in the academic, say either you need to have a lot of funding coming from the different government agencies or funding agencies, or you need to get in collaboration with the company who are ready to fund you, who are interested in something, what you are developing. Like if you are developing some drug and you have got very good results, there are companies who associate with you, they'll give you funding. They will also guide you to go through the development pathway, and of course they are companies, they have much more experience and all the required necessary knowledge on that. So that helps a lot. So that can be done. But if you do not have that much of money or that level of collaboration, you can definitely get some consultancy hired, get some expert advice for that particular part, for that particular slot of difficulties, and then move to the next step, and then again do the same thing, then move to the next step. But definitely you will need a lot of money doing this, so that needs to be assured that from where the funding is going to come.
So, thank you once again for your, the very experienced insights, I would say, okay, on this. And I think your consultancy services, or, these kind of ventures, what you have done is an eye opener for all the participants here, okay, who are associated for at different levels and with different places, who can definitely take the your expertise and your help for the further guidance of their the 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 drug discovery process. One last thing in that: do regulatory agencies recognized an academic setup
No.
environment?
No. So in the beginning, when you're providing your discovery outcome, mechanism of action, your animal study data, those kind of data are accepted. But you definitely need to have the record of them, like your laboratory notebooks needs to be completed. You are having some good laboratory practices, like the research is overseen by the head of the department, or the assistant professor, whoever is the lab head, should be looking into the data. So the overall clean environment in terms of data, clean environment should be there, complete recording should be there, and that's what is required. And then, but then when you go to the next step after discovery, you need to have GLP setup, which is there are the particular setup required as per the regulatory, and which is again requires money. It's not like that academic lab cannot do that. They can do that, but it's a lot of money associated, a lot of people to maintain that kind of facility, having instrument qualified, then logbooks for every instrument, the maintenance happening time to time, calibration happening every time you perform experiments. So those are the requirements, just to make sure that the data coming out is fully authentic and fully recorded and can be seen anytime. So it can be done, but it's more challenging. But yes, up to discovery level, the all data are accepted as long as they are recorded in well-mannered.
So thank you, Dr. Swati, again for your insights on that. Okay, and I think we are all really grateful to you for for these pearls of wisdom. And once again, from the entire team here, we are really grateful to you. Thank you for joining, and we hope to collaborate with you again in the future. Thank you.
Thank you. I look forward to work with all of you. Whoever is interested, I'll be very happy to talk to you, and we can always have a call. I'm from India, so I can come India whenever I can come. I can visit. So it's not in deal breakers.
Okay,
So, yeah.
and
Thank you.
just
Thanks, everyone.
just to let know everyone, as I know a couple of them here, including Dr. Swati, she's very helpful in person. Okay, so whatever you are trying to, if you go with her any kind of problem, you will get some kind of solution. So I have already put her email in the chat. So kindly connect with her, okay, if possible. Thank you once again. Okay.
Thank you, Debashish. Thanks, everyone.
Okay, so before moving on to the next session, I just wanted to announce a couple of things. Number one is that, like yesterday, we will share soon here in the chat both the attendance link and the feedback link for you to fill kindly. Please look at that and fill those. The attendance link, as I think now you all know, will be shared here only in the chat, whereas the feedback will be shared also in the WhatsApp group. Okay, for some kind of limitations, we cannot share the attendance link in the WhatsApp group. So please have a look at that and kindly fill both of it. Okay, though for your convenience, as already said to you, we'll keep giving reminders to you, okay, in this session itself. Next, a very important one, and I think maybe you're all a little concerned or looking to it, the evaluation test that we are going to conduct tomorrow, after the technical sessions are over, okay, and before actually the valedictory session begins. So it's called the test and feedback session. So the test actually will consist of 50 MCQ questions, which is covered from, which is the questions are taken from all the sessions from day 1 to day 5. Okay, please note again, the day 6 sessions questions will not be included in the test, okay, because we know that it might not be easy for you to go through those material in the same day. Okay, so again, I'm repeating for your convenience, the test tomorrow will consist of 50 MCQ questions, totally of you have 50 minutes to answer those, and each question is of 2 marks. So totally the test will be of 100 marks. So 50 question, 50 minutes, 100 marks, okay, is the frame of it. And you need to submit so we will share the link of the test. The test will be in Google form. Okay, so the link, that means the Google form of the test, the link of the test rather, will be shared here in the Cisco chat itself. It will be not shared anywhere, including the WhatsApp group. So you have to connect to the link from this Cisco chat itself, complete the test, and please make sure that you submit it correctly. You can please keep a watch of your internet connections and all, because if you don't submit the questions, we will not be, we will not have your answers. Okay, so I hope you're clear with it. Any doubt, anything you can ask
It's
us.
a one-time response, sir. No editing can be done.
Yeah. So that's one important thing, as madam said, it's only one-time response. Okay, so you need to think a little bit, and you can attempt those questions. Okay, don't worry. Okay, I think all of you can very easily answer all the questions. Okay, I hope you're clear with it. If you have any further doubt, please let us know. We'll again, we will make everything again, or repeat everything again. So with this, I like to hand over this session to my respected co-coordinator, Dr. Moshmi ma'am. And ma'am, you can kindly take over from here on.
Thank you. Thank you, sir, for the informations and sharing. Now, I, Dr. Moshmi Das, co-coordinator for this 6 days Atal FDP, and in this 5th day, would like to have the privilege to invite our next speaker, Dr. Abhijit Gorai. He is a senior project manager in Cognizant Life Science, Calcutta, West Bengal. Before his session start, I would like to share a few glimpses of his 19 years experience. So Dr. Abhijit Gorai is a BTech and MTech from West Bengal University Technology, Calcutta, and being a veteran of 19 years of experience in various sectors at biopharma R&D operation, life science, digitalizations, biopharma product development, QC, and regulatory filing at various capacities in organizations like Biocon, Reddy's Lab, Pfizer, Lupin Pharma. He has also a vast experience in various sectors like characterization of functional domain of the biosimilars, biologics, and currently his focus is on the biopharma domain. With this short glimpse of his vast career, I hope I have justified his career profile. I invite once again Dr. Abhijit Gorai for his lecture on the topic entitled Biopharma R&D and Automation Opportunities. Biopharma R&D and Automation Opportunities. Sir, the floor is yours. You can take the floor for giving us some deep insights on this particular topic. पार्थ भाई, अभिजीत सर को प्रेजेंटेशन राइट्स दे दीजिए. सर, आपको प्रेजेंटेशन राइट्स दिया गया है. काइंडली चेक सर, आप शेयर कर सकते हैं अपना पावर पॉइंट.
Hello everyone. A very good evening, good afternoon, or good morning if anywhere abroad. So thank you for inviting to the session. And so am I audible, and is my screen visible?
Yes sir, you are audible and your screen is also visible.
Right. Okay.
Sir, kindly make the slide full slide.
Yeah, let me try. So is it in the full slide view?
Not yet, sir.
Dr. Avijit, if there is any problem, in for making this making it full slide, you can just increase the magnification of it. Yeah, I think
this is good. It should go.
yes,
All right.
yes, it's fine.
All
Yeah.
right. Okay. All right. So thank you. Thanks, Dr. Devashish and the team, as Devashish, to invite me. And it's a pleasure for me to present in such a prestigious session for the Faculty Development Program. So many thanks to the organizing committee. All right, so I would be talking about the biofarm research and development and automation opportunities. So, thanks, madam, for introducing. So, I'll just talk about a little bit more. So, I basically come from biopharmaceutical research and development for many years, where I have worked for different biopharma companies like, Biocon, Dr. Reddy's, Pfizer, Lupin, these kind of companies into drug discovery space for novel biologics as well as the biosimilars drugs. Many years have been worked on the mostly on the functional characterization of large molecule. So when I say functional characterization means basically biologics drug development in the space mechanism of actions of drug. That space I've worked for many years, and then with that biotechnology product development and understanding from end to end, I moved from biotech to the digitalization side of it and currently work for Cognizant, where I work on the digital automation space for the biopharma on the discovery research, and that's the about myself. So I would like to also thank my past speaker, Dr. Swati Patel, for the wonderful background to the biopharma space, different stages of drug development, and then from there to the on the research work that she has done as a past career and currently what she is doing. Many thanks to that. So with that note, I would get into a little bit into more on the biopharma journey. She had covered mostly on the biopharma drug development space in the beginning of our presentation, where I would get into more detail would be on the biopharma research and development, which is a segment of the entire product development flow. So it actually collects the earlier presentation and further detailed insights to the research and development space in the biopharma area. So my flow of the topic would be, drug development flow overall, and then into R&D, and then I'll spend some time on the analytical and functional and structural characterization. Then we'll talk about the product life cycle in biopharma space, and then automation opportunities. And I'll definitely share my experience as a part of my entire past 19 years of journey. All right, so as we know, so pharmaceutical is very common. What we are very known to the pharmaceutical drugs. These are the chemistry-driven drugs. So let me introduce from the core, what is biopharma? So basically, biopharmaceutical is a pharmaceutical which is derived from the biological sources. It is basically from the origin, starts from the cell culture, and that's where these cells start developing these drug molecules inside the cell, unlike the chemistry or pharmaceutical drugs. That's where the biotechnology drug development is the core biotechnology development starts. So as we have heard, so all these vaccines is nothing new, right? It's a very old technology in the vaccine therapy has already been there for pretty old in the journey of the drugs. So again, so vaccines is again a part of biopharm, and then NB is something new, which is the novel biologics come into place when it is actually developed inside the cell. Biosimilar is like a more like a kind of a generic. We could think of the novel drug is there, and we are trying to make a similar copy of the novel drug. That's where biosimilar business come about. So this India is mostly focused on the biosimilar business. Yeah, novel there is a presence, but more than that, a huge billion-dollar business in India and Asia Pacific is into biosimilar drug development, which is currently going on. Apart from this, other part is the cell and gene therapy products. Swati has earlier explained on the cell therapy in the T cell therapy space. That's the similar one as what I'm talking about. Now let's get to the biopharm drug development as a process. So what happens? It starts with the discovery phase, where we actually look for the potential lead, and we call it as a lead identification, and then lead optimization at the discovery phase that eventually ends out to be a potential drug candidate for further development. And then starts the preclinical and the preclinical research, where we actually understand the molecule structurally and functionally and try to develop a process to develop the molecule. So one way we actually develop a process by which we develop that medicine. We actually work on to prepare the medicine. On the other side, in the preclinical, we work on the characterization of the molecule, whether they are structurally good or functionally good, how it looks like. So that's the on the stage of the characterization studies. So one way is the process development that happens in preclinical, another way is the characterization assays to understand the molecule. So and then once we have the initial level up, preclinical studies done, we have some safety efficacy data available, we go for the IND filing, which is an investigational new drug, or it's in case of in B, or in case of biosimilar. Again, we go to the regulator and demonstrate that this is our initial data looks like, and this is what we would be actually developing at the very early stage of kind of the first stage of the preclinical level, and we take a regulatory approval and then go for the um, clinical material development at the different phases. First is phase 1 clinical trials material, then phase 2, then phase 3, and then finally at this stage, which is the phase 4, the final level. And that's about the clinical trials journey, where the clinical material of phase 1 being made in the pilot plant, in the manufacturing facility, and then that gets to clinic, and then they are tested for the human studies and all. So and it's a long journey for coming from the initial R&D laboratory to bring it to the pilot plant, to the finally to the manufacturing facility to make clinical material. So we will go further detail, and then after that, once we have a data package, we go for the filing. So they are the different regulators we go for. US FDA, EMA is Europe, PMD is Japan. So we file the entire data package and look for marketing authorization approval. So after we receive approval, there's a long review process for these regulatory approvals. Post that, once the approval is received, then the companies are ready for manufacturing these drugs at the large scale. We call it as a commercial scale development, and then it goes to market. That's the entire journey overall. And now, out of which we will focus more towards and the biosimilar drug development at the preclinical and early discovery research. And that's the topic today, where we'll talk about the R&D and we'll explain the automation opportunities, which are the new area which are coming up to make the bench to bedside much quicker, turnaround time, efficacy, and that's where the automation comes into the picture. With this note, let's go to the product development flow or the drug development flow in the R&D. So what happens, as we actually said, it's in the cell. So basically it starts from the cell line, where we know we in the in the universities, and we teach a lot of animal cell culture, immunology, immune therapy, and then cell based cell culture work. So that's where it starts. When we have the clone development or the cell line engineering is the first workflow, where we actually do the inceptions, we do the gene transfection, and then finally we actually try to develop a clone that we want to develop it further. So I will get into more details in the later slides, where we have one slide for each of these area. But to oversee or overview on the entire topic, what happens in the biosimilar research and development flow, it starts with the clone development. So it's a process where basically we insert a gene in the cell, mostly Chinese hamster ovarian cell line, which are used, and they start expressing the outcome of the gene, the protein, which is the drug of interest. And then when we identify a right clone in the stage of the clone development, which is a long process, and then we use that clone to further upstream level, where we actually try to scale it up so that it starts making a larger amount of the drug molecule. And that's the we call it as a upstream culture in the fermenter. That's where we actually start making the drug in a larger volume. at the R&D scale and try to optimize the nutrients, optimize the feed so that the intended drug molecule is actually received or kind of produced in the bioreactor. And then we go to the downstream purification process where we actually look for the purification of this drug molecule in the downstream, where we try to purify our intended protein of interest, which actually we started to looking for at the very beginning. And then finally we make a drug substance and the drug product, which are at the stage when we make a vial of these molecule, and then that's the kind of the final level of the R&D level. And once this process is optimized, that gets transferred to the pilot plant before manufacturing. So pilot plant is a scale up happens for the upstream and downstream this process, where larger amount of the drug being prepared, which is a large scale fermenters come into picture, and then further scale up happens in the manufacturing facility. And what goes hand in hand from the beginning is the bioanalytical characterization. This is about the one sided is the process development. On the other side, what drug we are making, that actually needs to be characterized. That's we call bioanalytical characterization. We do series of assays like HPLCs and mass spectrometry, different kind of assays to characterize the molecule structurally, how good it's at. And at the same time, we do a series of functional characterization assays to look at the potency of the drug. So how good will it work in the human body once they are treated with. We do a huge level of cell culture based assays and cell based assays to understand the functionality of the drug molecule. So this is about the journey in the research and development. And a typical biosimilar drug from initially from the inception to market, it would easily take 7, 8, 9 years for our entire drug development journey. Whereas a novel biologics, it could actually go for many years, 10, 15, 20 years, based on the discovery level research, which are much intense to identify a candidate, and then identifying the right efficacy through clinical trials and all. It takes a lot of time. So that's why the discovery NBs take a lot of more time than the biosimilars. So with this, let's get to the a little bit more details into the clone development. What happens? We had already discussed that what happens in clone development, but let's look into a little bit more detail on the first stage of the biopharma space for the research and development. So in case of clone development, what happens? We have already, I mentioned about the Chinese hamster ovarian cell line. That's where we do the transfection, and then we get the gene of interest getting expressed. Once it starts generating the once this cell, after this is this transfection, and then it starts producing the protein. We first do the limit dilution steps. So basically, this is a step for how do you basically grow from a small scale, first in a small plate to a 96 well plate to a 12 well plate to 24 well plate to 12 well plate to 6 well plate. So this is how you actually starts growing in, and then you get to the different culture flasks, small scale culture flask. So what is important in clone development? So one way, when you have the intended protein is coming out from the cells, we have to get this protein, understand that what is the protein titer which is coming out. So is it really a suitable amount of protein which is coming out? So we do a lot of, in this stage, two things happens. First is we need to ensure that it is coming from a single cell. Monoclonality is something which is very important at this stage. There are different technologies. In the flow cytometry is one of the technology where we do a single cell level understanding and where it is a monoclone. There are imaging platforms which are available on today. We look at them and to see that it is coming from the single cell. That's where the monoclonality is important, is the first step. And when we see it's coming out from a single cell, that means ideally a 96 well plate. When we do a limit dilution, its expectation is in a well, one single cell is deposited, and that single cell is getting cultured and making clones, and then in the colonies, and then finally it is producing the drugs. And once it is in the sec flask, in this step, we need to collect the supernatant from the sec flask and then harvest the supernatant, and then you need to do a IgG titer ELISA to see what is the IgG level, the ELISA titer. So, and then finally you need to look at this stage what is the which cell or which clone which is representative from coming of the one particular single well in the 96 well plate is actually giving a high value of the drug through an ELISA titer. So that's how we identify and shortlist or screen a first level of 5, 6, 7 clones, which are the good performing at the clone development. They become the potential finally clones, which would be further um, the precursors for the producing the large level of the drug molecules in the further bioreactors. So that's how first we need to identify here at this stage some of the clones, and then those clones are there's a bank which are prepared from the single clone and single cell, and then that's fairly transferred to the upstream level for culturing it at a higher level in the bioreactors. So now let's get to the upstream and the downstream further. So what happens? So now, as we already seen, what happens in clone development, right? So now these clones are further cultured in the upstream where we have fermenters further one by one. The fermenters you're seeing in the picture that you first need to do a huge level of optimization and media and optimization of feed, optimization of the whatever the growth components inside the fermenters, and so that you see that how the cells are growing here and in which conditions need to see that we are receiving the highest level of the titer of that means the highest level of the drugs are coming out in these particular fermenter conditions, and there are multiple fermenters run in parallel and then need to collect the supernatant from them and look at the titer from them. So once now these are, as we said, we started from the cell line, and these Chinese ovarian hamster CHO cell lines, what we actually use, not only this, you will get the gene specific, the protein outcome, it will also get some of the additional unwanted stuffs coming out in the harvest. That's where you need to first do the purification process in the downstream. It's a series of purification process that happens in the downstream operations one by one, and end of it we look at a purified drug at the end of the downstream process. That's where the entire flow gets into a different type of chromatography techniques that's taken into, and different type of filtration gets imparted, and then finally you try to get into the final purified product at the end of the downstream. So now let's look into these steps a little bit more detail, kind of a summary that we had seen in the earliest slides, the clone development and then upstream and downstream, and also to revise what we had actually looked into in the last 10 minutes or so. So we started with a gene of interest. That's for an example, let me take an example. The rituximab is an anti-cancer, anti-CD20 drug treated for, used to treat for B cell lymphoma. This binds with the CD20 receptors on B cells. So now you have a gene of interest. So now you have to do this transfection, which would actually essentially produce your intended drug, rituximab. So now you do this transfection of interest, and then it takes the gene uptake and do all these molecular biology experiments you do at this level for this gene delivery, and then finally you do the whatever when the drug is coming out, you do the clone cell isolation, and then you look for the titer and then look for the clone expression levels, and then finally you prepare a stock when you have identified the particular titer of the drug, or what are the kind of the shortlisted 10 top for performing clones. That means these particular clones are now ready to further pull it to the upstream level for scale up. So again, you make a bank here, and then you go to the upstream and then revive this particular cell line, and then do a scale up level culture. First level 1, then level 2, and then do a viral clearance. And then once you have this upstream scale up is done, and then you get to the downstream purification process, where you have first the harvest and then the bulk drug, feed praise, and then the primary culture and this buffer exchange, and then purity, as I was just explaining in the last slide. There is a series of centrifugation and the filtration procedures by which you try to intently get the final outcome of the drug that we started at the early clone development. So that's about the process development. So basically you start with a clone and then you do the upstream and then downstream is the further process that you develop, and these process actually gets transferred to the um, pilot plant. Pilot plant is just before the manufacturing, where you scale up your first whatever is done in the small scale R&D, that you scale up to make a larger volume, and then finally it gets to the manufacturing plant. So in parallel, what it runs is the huge workflow for this characterization. One way we do the process characterization, and the other side we do the product characterization. Product characterization is nothing but actually ensure the drug molecule is actually developed for the intended purpose, and they present the right structure and they have the right functionality. So now process characterization, if we look at, so we have already discussed earlier that we have a clone development and then we have upstream and downstream. So these upstream and downstream, in the fermenter level process is what is actually developed. The same process is actually transferred to the pilot plant. So that process characterization is very important at this stage, so that the right process is transferred and then it's further taken up. On the other side, the product characterization that we talked about, so now there are primary structure, secondary structure, tertiary structure. All these are looked into different kind of HPLC as well as mass spectrometry or CD or different kind of or fluorescence or different kind of techniques. These are the standard techniques which are used across in different industries to characterize the molecule structurally. At the same time, we do a lot of functional characterization at this stage. If you have apparently seen some of this kind of the potency assays Dr. Shwati was explaining in the last presentation. So the same thing is what we do, a functional characterization of large molecule. We do a series of assays in the lab to understand how the molecule is functionally active. And then we also have a bunch of assays to look at the impurities. What are the other impurities which has come in the process of this journey, because these cells are producing protein, they are producing some of the unwanted proteins also. So you need to ensure that it is not impacting your drug. Your final drug is actually does not have contain any impurity. That's all testing is done and clarified over here. So that's the characterization journey. I'll spend some time more on the functional characterization. That's actually, I've worked for many years in this space. So how do you do a functional characterization of drug molecule? So functional characterization is nothing but identifying the mechanism of action of drug. So earlier I talked about, I took a name called rituximab. So just a random example to explain so that it'll be easier for us to understand. So it's an anti-CD20 molecule, and rituximab works in the space for the immunological environment. It demonstrates CDC, CDC, ADCC, and apoptosis. So now, how do you do these kind of assays in the lab? So now, as we know, it's a CD20. I explained earlier, it binds to the CD20 receptor. So you develop a cell line based assay in the lab to demonstrate this molecule is actually doing the right cytotoxicity in the laboratory environment. So you develop such assays where you actually treat the cells, B cell lines, you treat this rituximab and demonstrate that it triggers complement dependent cytotoxicity. In the lab, you triggered these B cells with rituximab, incubate, and then the rituximab binds to the cell line, and then treat them with the complement. It could be the complement is either it could be from the human blood, you isolate serum and put the complement into it, or baby rabbit complement is another scenario which are used to demonstrate this is showing a cytotoxicity and the complement dependent cellular cytotoxicity. It also triggers ADCC. ADCC is a classical platform where antigen dependent cellular cytotoxicity. It's a PBMC based. So in the PBMC based scenario, it actually works in the, you collect the human healthy individual sample and look at the, ice from the blood. You isolate the peripheral blood mononuclear cells, and those cells either you culture in the lab or preserve it, or maybe you freeze it and use those PBMCs to treat with this rituximab drug. And it actually, there is a Fc gamma receptors which are actually triggered in this kind of the killing mechanism. This rituximab goes and binds to these scenarios, and this Fc gamma receptors mediated killing is happens. So essentially, we see the PBMCs getting killed. So that's how these assays are designed. So another approach for ADCC is engineered cell lines. So instead of PBMCs, sometimes the Jurkat cell known to express these RC gamma receptors, we take up those Jurkat cells, engineered them, put these rituximab along with it, and then we demonstrate in the lab that it actually triggers the certain level of these antigen dependent cellular cytotoxicity. This is a classic example. So there are many different kind of assays in the lab, where we actually understand how the molecule works. There are a lot of assays which are nowadays coming with the um, um, engineered cell-based assays, where basically we are expressing the particular gene of interest so that a drug binds higher level and then it goes and shows its efficacy. So this is how the functional characterization workflow goes in the lab. So it's a long journey to establish a drug molecule's mechanism of action, how it actually works in the lab scenario. So this entire journey works throughout the whatever the drugs are developed as a part of the upstream downstream to look at the what's coming out, and when the final stage of the drug is developed, that's where this functional characterization is mostly deployed to understand the potency of drug molecule. And then the standard assays are actually further when the molecule goes to pilot plant, then to the manufacturing, all the samples are tested using the final qualified assays in the R&D, or the assays are also transferred to the quality control where they're validated. Those validated assays are used in the quality control in the GMP space to release this kind of molecule batches from the manufacturing facility. So that's the importance of the functional characterizations. It is one of the very critical battery of tests in the specification of any drug molecule, what is actually released from the quality control and the GMP facility. So we talked about, in line with our today's discussion, biopharma R&D, what happens. We talked about the process development, we talked about structural characterization, we talked about functional characterization. Now let's look into the overall the assay development flow in the context of overall biopharma with a focus to the R&D level. So when we say assay development, basically, it's a case study of the analytical methods or the bioanalytical assays that are actually developed at the preclinical and discovery stage for several years. So they are basically the assays which characterize the molecule. So these are the R&D data package which are used for characterizing the molecule, and these data packages submitted to the regulators for the initial level of approval. And the later stages, all these actually get into the part of the regulatory filing when we do a complete characterization. So when we do the regulatory filing, one way we have the entire process oriented data package, other is the characterization data package. When the final molecule comes from the phase 3 trials, and those one-sided, those molecules batch release data are generated from the quality control in the GMP space, whereas the characterization data for those batches are generated from the R&D. So by that time, these particular methods should be a qualified method in R&D. They are actually used across at this end stage also to characterize the molecule, as well as those assays are transferred to the GMP. They are validated over there. They are used for releasing those batches. So that's the entire flow goes. The characterization assays actually lie in the preclinical stage, whereas the final assays gets qualified and validated when the molecule moves towards phase 2 to phase 3. So those assays are actually used in the GMP space. Those same R&D methods which are actually transferred to the GMP, but when the molecule is a kind of the phase 2 to phase 3 stage. So this is a very good concept to know. So you have the R&D level assays, they are first developed, then they are qualified, then they are validated. So when we have the molecule moving from clinical trials phase 1 to 2 to 3, we must have these mass assays validated in the GMP space, so that when manufacturing facility releases these samples for testing to the quality facility, quality control lab should have those validated assays available. So those assays are used as a battery of tests for releasing those batches. At the same time, R&D uses same assays at the qualified stage to characterize the same molecule which are coming from manufacturing facilities. So this is where the entire data package gets generated to characterize a molecule as well as the batch release. So this is how the entire flow goes. That becomes the part of the filing for the regulatory filing when it gets into. Now with this, let's get to a little bit introduction on the analytical method qualification. I have already covered this a portion, how it goes. So in the R&D stage, we have an early stage, we have a lot of methods which are developed and qualified. When I say method development, means you actually developed such an assay. For an example, let's say a CDC assay we have developed. When we say qualification, qualification means to test these assays multiple times, same drug, in respect to another reference standard, which is nothing but the same drug, and repeat it multiple times and ensure that it is developed and it is recovering to the same level of intended potency. For an example, if I run a 100% sample against a 100% sample, it should give a kind of the potency 1. That means it's very much comparable. So the method should be capable of identifying a molecule for an example in a case study 70 to 130%. That means my drug which is coming out from the facility should be capable in the stage of the either if I have a standard reference of 100%, if I have a standard reference of 100%, whereas I should have alternate if I have a standard reference of 100%, so my drug could be 70% potent or 130% potent. That's my acceptable range. But if it is below 70%, that means it is not capable of eliciting the same mechanism of action, same response. It is rejected in the system. So it is not accepted as a proper, potent, acceptable drug for release. So that's where qualification is very important to establish the method accuracy and precision. So accuracy and precision, these two terms are very important in the qualification space in the R&D. So basically, it ensures the method is actually qualifying the right drug which are coming out from the facility. So this is what happens in the R&D space. It's called qualification. The same thing with more rigorous, say, with more rigorous scenarios within the GMP facility that we call is a validation. So these methods are validated within the GMP space. So this is the flow. So first for the biosimilar space, first when you have a, say for an example, I give an example again. Rituximab is an example. I have a standard drug available. I'm making a biosimilar for it. So I have this rituximab. I have a reference standard. We call it the development reference standard against which I compare my internal in-house drugs that I'm making. So we call it the developmental reference standard. Then we have the interim reference standard of different categories, and we compare the drugs which are coming out from the manufacturing facility against these standards over a period of time, and then we demonstrate it is suitable for intended purpose. And that's what is actually happens, and it goes into a part of the regulatory filing, how the qualification and the how the internal reference standard qualification happens over a period of time in the R&D and the GMP space. So with this, let's get into a little bit now. I actually covered most of the R&D related activities as a whole. Now let's get to the little bit touch upon on the commercialization space. So now we have the all these R&D workflow, and then in it finally gets to the manufacturing on the operations facility. One way the methods gets transferred to the quality control, whereas the process gets transferred to pilot plant and then further to manufacturing facility. And manufacturing facility actually develops these drugs which are actually tested, and then finally it launched and then it gets to the market. So now this is where it goes, and then post marketing we have the surveillance and the patient support. There's drug safety, pharmacovigilance, all these things come into the picture. And so these are the entire workflow. We have on-site sales and marketing, and then market access, and these are the kind of medical affairs and that space of the post-launch what happens. So now let me get back to the overall the product life cycle space into to again to recap and to introduce the as a whole where R&D is positioned and how the entire product life cycle gets into. So we talked about preclinical space where we have the we talked about different methods. We talked about different methods and we talked about the uh, phase one, phase two, phase three clinical trials, how they are interlinked, and then finally it gets to the filing and launch. So in parallel, what happens in the workflow? So this is the now. Let me talk about the how the entire life cycle actually the product development gets designed into. So there is something called the QTTP, or which is the Quality Target Product Profile Assessment, which is at the very beginning. Excuse me. Alternate number তে call করুন আমি মিটিং এ আছি alternate number তে call করুন sorry so okay, I was talking about the quality target product profile QTTP. So QTTP is basically at the stage in the Swati talked about in the earlier presentation of the CMC, chemistry, manufacturing and control. See, the CMC starts from the day one of the CMC starts from the day one of the drug discovery, where we have actually the quality target profile identified. So what kind of actually quality drug we are going to make of the entire journey, and then from this QTTP, it actually gets matured to the assays when it gets developed with the governing strategy which is actually defined. So regulators are very critical to see how the entire QTTP or the product quality target profile was defined at the very beginning, and how it is actually product development moved to the entire period of time as a part of the CMC portfolio. That again gets into the a journey of the entire regulatory filing. So CMC is nothing but a governing strategy that governs from the R&D stage, from clone development to process development to the finally moving the molecule to the pilot plant and then to the manufacturing quality. On the other side, it governs the entire workflow from the day one, and how the methods have moved, how the documentation has been done, how the entire product development portfolio has been managed. So this is where very critical now digitalization opportunities are coming into under the CMC portfolio. So with this note, let me move towards the digitalization part, which is again our today's topic, the R&D and digitalization opportunities. All right. So before that, again, a kind of the explanation to the timelines and cost. So reference biological development timeline, and see, we talked about in the last half an hour, we are talking about the drug development journey. It actually looks for the in a discovery space, it is $1 billion business. And in the biosimilar space, it is much lesser, but it's again in terms of the $1 million, and it goes for many years. It in biosimilar, it typically 7, 8, 9 years, whereas discovery it takes 12, 13, 14 years of the journey. And this involves huge cost, huge timeline. So with this all these things now, and this entire journey has a lot of manual operations, this entire journey has a lot of manual operations, manual interventions, where people are actually dealing with a lot of lab notebook, manual data recording, a kind of the recording of the experimental data. And these experimental data are lying in one experiment but is not connected to the second experiment. And then we have probably a journey of 7 or 8 years when we file this drug to the regulatory market. Regulators are asking multiple questions. Documents are not ready because 10 years back, one scientist has done some assays in the lab, which is somehow not written completely or partially written. Then some of the data has been used from there for the regulatory filing, but that has no proper traceability. Alcoa principle is not maintained. So that is where all these, quality management system coming into picture. And then with that, now today's era, to move away from this manual journey, a lot of automation related stuffs are coming into the picture. So now with this, let me introduce what automation is all about. So now when we say automation in discovery research, so now as we know classically now, this discovery research is a core bioinformatics driven, a lot of structural and a lot of, we look for the different possible combination of markers and different things, right? And then we have we have classical methods of docking to look for the different combinations and what could be the potential marker and all these. So in this place now, artificial intelligence is coming out with a big way, so which we call as an AIDD space. That's one of the area. Second is automation in the process flow for the research and development, manufacturing, and quality control. So when you say R&D, I will talk about more on the R&D workflow, but just to say the manufacturing is another one huge workplace where a lot of automation opportunities are coming in, because online data recording system is one of the key critical regulatory attributes which are coming up. So now companies are moving from paper to online, which is a lot of electronic lab notebook, laboratory management system, LIMS, and all these things are coming out to move away from the manual system to the automation. And the last important point is the documentation. So entire documents, ELN and lab notebook is one example of automation in the documentation, but there are many different approaches for the documentation portfolio for managing. How do you see, if you have to think of a journey of 10 different years, and where people have done a lot of manual work and a lot of scientists have worked and people have come to an organization, developed something, and then moved different people's involvement. So entire manual documentation is now getting into a digitized node. So that's where the automation of in the documentation portfolio is coming up. Now let's get to a little bit more detail. So what is AIDD? So artificial intelligence driven drug discovery is a kind of drug discovery new area which are booming up, where a lot of algorithms are coming into, which is capable of predicting, which is capable of predicting the right bonding, or what could be the right interaction of these different molecules, or what could be the potential markers. And this phase, a lot of artificial intelligence driven work is currently coming up. So that's one potential area where we actually we see a lot of opportunities are coming up in terms of the automation in the discovery research. There are different platforms, different companies are coming up. A lot of software companies are getting into collaborative work or the interactive work for the core discovery, giant pharma and biopharma. That's where all these AI driven approaches are being taken up. Second is let's talk about the early stage research. So coming to the couple of spaces. So how do we fast track? See, one way is the in the lab we talked about the process development research. We talked about the biopharma space, analytical research. We talked about the functional characterization research. Now all are, one aspect is the manual operations, where we are managing everything in the lab notebook. That moving that to the online platform, which is the electronic lab notebook, is one sided to it. Other way is AI driven approaches. So let's say I do 100 different assays in the lab to predict to reach to a certain conclusion. Can I do 10 different assays instead of 100 and predict my outcome, what was being actually established from 100 different assays before? So it helps me two ways. One is it saves time for 100 different assays versus time for 10 different assays. Second is the precision. While doing 100 different assays, I've done a lot of manual workflow. I might have done a lot of manual errors. People might have done a lot of manual unintentional unknown errors, which has impacted the decision. Whereas when we have, let's say, past I have 1,000 different assay points, I use that data, make a predictive model and through AI, and then I use my 10 different current experimental outcome, and can I predict my outcome, what would actually come after 100 assays. So that's where it's coming into AI for this drug discovery and early stage research and development space. Last but not the least is the laboratory automation and modelization in line with lab of the future. I think many of you must have heard lab of the future is a new concept. We call it as a industry X.0, 4.0, and I think currently we are moving towards 5.0. It is nothing but a new concept where we are trying to digitize the entire workflow. So imagine a scenario. There used to be 10 different let me take an example of HPLC, 10 different HPLCs in the lab, and those HPLCs were being manually managed or manually run through, and those HPLC data and chromatograms are being stored locally in those PCs. Now, when scientists are compiling or looking at the data, they need to go to each of them, extract the data and pull it together, and then interpret it. So imagine a scenario where 10 different HPLCs are connected to a single platform. One way, you are operating those HPLCs online through electronic platforms like Yale and LIMS. You are giving commands to capture the data over there. On the other hand, you are extracting the data from HPLCs through to in a centralized database or centralized repository in a large scale. It called as a data lake in the classical IT terms, where you actually see all these data coming to a synchronized way and harmonized way, and then scientists and analysts can look at those data in the different fashions, and then like give you an example of 10 different HPLCs. Now think of 100 different instruments across 10 different labs in R&D. If the senior organizational leader wants to see everything in one platform, you don't need to go to the lab. You would be actually seeing with some comments in the automation platform. You would see all the data put together in one area, and then you will be seeing the interpretation of data also through some of the AI aspects and all, and then finally you actually get a better precise and visualization of the data through this kind of lab automation and modernization aspects. So lab of the future is a concept where you actually see, a lot of, in one way, the automation aspect is, now we must have seen that robots are coming to the lab. They are walking, or some of the drug dilutions are being replaced by robotic arms, which is standard. On the other side, I think the future lab is coming where actually robots would be working in the lab. You would be just giving the comments to robots what to do, and they will actually do the work in the lab, and then this data would be generated in different instruments, and these instruments will actually get connected to a central repository where you will see all the data, and finally you'll see the package in your hand, which is the lab of the future or the future lab further in the concept which are coming into. So digital automation is looking towards the next stage of the discovery research, where not only we are moving from paper to the automation, on the other hand, we are implementing different ways of managing data, different ways of implementing data, visualizing data, and on the other side, we are able to manage equipments and instruments in a different way in a larger automation driven platform where we are able to manage the entire thing. So I already talked about ELN and LIMS. So last but not the least, in regulatory filing. So I already took up that example before. Let's say regulators are asking 10 different questions, out of which couple of questions are related to the early discovery research of the molecule characterization. That data is there of 10 years before, which was done in the lab. So how would you retrieve that data quickly in a short time when the regulatory filing happens, and after that you have a clock starts? Within 120 days, you have to respond. Depends regulator to regulator. So now how will you manage? So now all this documentation process is getting online, so that, and not only that you retrieve those information, but also you actually get all those information in a documented format. Let's say, now AI driven search engine, I can ask my my chatbot or my AI search engine, can you generate a report using XYZ data sets? It does. It gives the data. So imagine a scenario, a manual scientists are looking for this data for a few days, and then they are preparing a document, then they are reviewing it. It's in one week story. These AI driven options and opportunities would be taking hardly few hours, or 1 week versus 1 hour. That's the fast tracking, or that's the speed, that's the precision. This is what we're talking about. So anyway, this is about the journey of the automation or digital automation we talk about. I covered, mostly the R&D focus, but in the manufacturing space is much more larger. There is something called digital twins are coming up. So the entire manufacturing operations will be operating from your lab, or from the office workspace, and you would see what is happening in the parameter level. That's where the digital twins are coming up. Another example to add on. All right. So I think we covered most of our topic. Our focus was the biopharma overall understanding the product development portfolio, and then into the focus what happens into the research and development, and then further what are the opportunities for digital automation, and where are the challenges, and where are we moving forward. So with this note, I think mostly I've covered most of the now last topic I have. So let me share some of my past experience. This is especially when I was working in Lupin, an experience for Nipexto is the biosimilar for etanercept. So I had an opportunity to work with this drug to share my experience. So we had developed this molecule, and when I took up this project, I think it was a little at the late stage. There was a lot of historical data which are already being filed, and then after filing the data, what we it was a regulatory query response. Let me explain how it actually goes. So we created a bunch of assays. One of them on the functional assays, understanding the apoptosis of the drug. So we filed it. I think this is probably, I'm talking about the EMA, Europe. Yes. And then the regulators came back saying that, yeah, this is fine. Mechanism of action is there, but we wanted to the molecule to demonstrate this apoptosis activity using this particular cell line because it has some specificity. And that's where your query response starts, and that's where your day counts. It's a 120 day you will have to respond. So then, and that's where the collaboration comes. This is a workflow for R&D, a quick method development, then transfer it to quality control. So you involve quality control personnel within the R&D so that they know what is happening to some extent. And then when the R&D person transfer to the quality people, then they start quickly, they do a validation, the qualified methods, they validate it. And then you, not only that, you kind of relook at the entire workflow at the same time. Whatever batches you have released maybe past couple of years, you again analyze those batches using this again developed method. So it's a kind of a live experience that we had through, and it was really challenging, but it was really great to see. I think what regulators are asking, they're scientifically quite rational, nice questions. So you are actually addressing those questions by doing a series of GMP work activities in the lab, and with the collaboration with different team. And so basically R&D, what you develop, that needs to be accepted by the quality control, right? And then this quality control has to kind of validate those methods, and then those methods has to be kind of used to release the batches. That's where the endpoint. So you again redevelop those particular package and then submit it to the regulator. So that's the journey that it's a kind of the experience to share when I was working with Lupin, and it's really great to see when you actually receive a kind of the regulatory approval. So it's a molecule that you have worked, and then it's going to the patients and benefiting lives. Is something that you have done for humanity. That's the kind of the accomplishment that you feel through when you actually reach that state. So I have kind of shared one from Lupin. So I have worked for different biopharma companies, and I've gathered my experience over a period of last 17, 18 years on that space. And with that, when currently I am into the digitalization space, where I am more focused into this entire discovery or the preclinical or clinical, the entire research flow, where all we can hook in different kind of automation opportunities, and where all something can be digitized or automated so that you get more precise data. At the same time, your time to market comes down, and at the same time you have more control way of operation in the entire biopharma development research and the entire portfolio. So that's where currently I'm working into. So the kind of this slide is to just give you a feel that how you actually do, and then also to showcase my to share the journey, and then what I've worked in this space, and then currently what I'm into. So with this, let me get to the towards the last slide of mine, where this is again from a paper. So most of the information that I've shared, apart from my experience, is the public domain information, nothing very specific. So from the different literature, I put it through. So I came across this picture. Is very nice. It's something very appealing. So what actually this research you do? So you cannot you put a seed in the soil, and you cannot get the fruit in a quick couple of months or years, right? It's a tree you are seeing at the end. It's a big tree which is giving you fruits, let's say mangoes or apple, whatever it is. And now I think of this, how much time this tree actually takes when you have planted a seed, when you have seeded into the soil. So that's the journey of entire biopharma research. You are actually touching human lives. You are starting from clone, where you are actually inserting a molecule inside the cell, inserting some gene inside the cell, and then it's creating your drug, and you are going through this journey of $1 million finally to make drugs, and when it takes 10 years or so, and then when the drugs are coming out from the manufacturing facility, you test it and then you release it to the market, something that rotten fruits you cannot sell, right? So something that you'll have to look at there, right, for the internet purpose, and then gets to the market. So that's the journey of biopharma we are currently into. So with this, now let me share a couple of snapshots, how the laboratory looks like. This is in the bioanalytical and the cell culture laboratory. So you see on the mid one, right, these are different HPLC system towards the end. But the down panel is right side of the plate reader. The plates are getting into, and the down panel you see the cell culture lab. And then in the top panel again you see a workbench where left side scientists are working. Right side again you see these HPLC ones. Again in the mid one left you see in the microscope scientists are looking into some of the cells or some of the cultures in the lab. That's how the real laboratory looks like. So next is to look at how the manufacturing facilities look like. So as I said, from R&D you go to the pilot plant, and then you'd go to the biotech manufacturing facilities are huge. So I think there are fermenters of several lakh liter. Think of, we start with a 1 liter, 2 liter, 10 liter, 50 liter, 500 liter, 1000 liter, 10,000 liter, and finally it goes to lakh liters, the capacity of such big fermenters. That's where these drug molecules are produced into a large scale. So that's the entire journey in the manufacturing process, when you actually keep upskilling and get to the manufacturing, and then finally these drugs are coming out to the patients. Okay, so that's the journey. So with this, yeah, I have used some of the references. They are public domain information, web resources, research articles, and I think some of the open information on the digitalization and my experience. So with this, let me thank to the organizing committee, to the team, and the listeners to my presentation. Many thanks for last 1 hour listening to it. And now I think I would open it for quick questions and then any other things that we can discuss so that it can benefit the faculty development program. I think I'm good with
Thank
it.
you, sir.
Thank you. And madam, please go ahead.
Thank you so much, sir. This is here of side Dr. Maushmi Das with my coordinator, Dr. Debashish Banerjee. Once again, thank you, sir, for the deep insights on this particular biopharmaceutical stages. And if I can make an excerpt of your this vast presentation with a few important glimpses which will be alluring us, like analytical characterization and the functional characterizations, flow of the biological assay during any product development, and especially what you have mentioned, that digitalization and its importance in regulatory affairs as well as filing documentations, how the AI tools are now getting ahead, and along with that your own experience during this journey, how you have felt during the product development and regulatory queries, and as well as a few of the things like Nefexto, biosimilars, and etanercept. So these things, sir, really cannot be finished up in one point of line. So I guess my participants and we all are thankful to you to just show in a span of short of time the entire picture of these biopharmaceutical industries, and behind the product drug development, so many struggles, and the kind of the tireless nights you people generally spend. Being a bioprocess practitioner, and I generally deal with the fermentation technology papered in the academics, some things really catches my eyes during the process of the manufacturing development. And it is so fascinating that if our students also get a chance as an intern, or like in as a part of the projects, maybe they can be more observing the insights of these struggles behind the product development skills. I hope my coordinator will also agree with the same, because he is also once upon a time in this field. So with this, sir, I once again acknowledge your presence with my team and my participants. Thank you from our side to make us aware of this particular domain and facets and areas. Once again, thank you, sir. Parth Bhai, can we have a round of applause, please, for this excellent presentation? sir. Now I just checked once the chat box for the couple of questions.
There are, ma'am. There are a few.
Yeah. So the one question which you can see right now in the chat box, that is, can you give me some light on some definition of market authorization of new drugs? Presently, are your R&D lab doing the AIDD?
Can you repeat the question, please? I think two questions got merged. Can you talk it once
Yeah.
again?
One question is, give light on some definition of market authorization
Okay, all
of the
right.
new drugs. That is the one question.
Okay. So MAA is the marketing approval authorization. So basically, this is nothing but, you have the entire drug development journey. After that, whatever you have done, it has to go to market, right? So what you actually look for the regulators is an approval. So regulators will give you the approval for marketing authorization. That we call it as a mark. It is a commercial terminology, MAA. So this marketing authorization approval is given by regulators. For an example, I have developed my entire drug. Let's say I am presenting a company. So my company has developed the drug, and this is the data package, and I have filed this data to the regulatory filing authorities. And then the regulators come, they do a series of inspection, and they do audits and then inspections, and then audits. They have some findings. They give up questions. That's why the regulatory query response I was referring to, and then you respond to those queries again. And finally, what you receive from the regulators is a marketing authorization approval. So that means now you will make these drugs at the commercial scale in your manufacturing facility, and you will sell these drugs in the market. So that's the MAA. So I hope it answers the question.
I hope Dr. Rupa Kumar has got his answer. His next question in the same line is, presently, are your R&D lab doing on this AIDD?
AIDD is coming into big time. Actually, there are most of the companies are getting into AIDD. I think Indian Biopharma is doing it into the novel drug discovery space to some extent. But the large biopharmas, for an example, Pfizer, GSK, Merck, Sanofi, global biopharma companies, Amgen, they are doing a huge level of the AI-driven uh, studies or AI-driven workflows to look at the what all the automations can be done, how predictions can be done, how do we minimize time, how do we make precise data in a shorter time. So that's is currently going on. In India, it's yes, but more than that, I think the large biopharma space is already into in a much larger domain as compared to the Indian biopharma. But we are definitely into it, very much into it, and that's the next level. That's the next future of this biopharma space, actually.
Thank you, sir. Dr. A. Madhavrani is asking a question, sir. Will you do clinical trials with humans, sir? If so, how do you select the community for the trials?
So I'll collate the answer to the context that Dr. Swati Patel was mentioning in the last presentation, that clinical trials, how do you do the clinical trials? Say, for an example, I think let me take an example for the last presentation. So she was referring to the T cell therapy diabetes cases, right? So now for an example, now let me generalize it. For an example, let's say it's a novel therapy which is coming into. So now we'll first look for a small scale clinical trial patients. At least if it's intended for diabetes, then you do a phase 1 clinical trials with very selected patients with placebo and the control normal healthy individual to see how these drugs are working. So how do you select? Yes, for an example, if your drug is intended for type 2 diabetes, so you will collaborate with hospital and select a set of type 2 diabetes patients with so and so criteria, which has to be mentioned in the protocol for clinical operations, has to be approved by clinical authorities. That's how you select. Once you have the first data sets, then you get to the phase 2 clinical trial, which is even larger. Let's say 40, 50, 60 clinic, even more, depends on the what kind of population. So now again you will have to do a clinical trial. For an example, you want to a classical or traditional approach is the territory you want to sell, you will have to demonstrate clinical trials data. You have to let's say you want to sell in Europe, so you will have to use subjects from Europe to demonstrate the safety and efficacy in Europe. But a lot of new things are coming up now. FDA or the EMA are accepting a different populations. Some of the hypothetical studies also they're accepting. But that's a different part. But to answer your question, so you need to be very focused on the drug target, and that's why you choose the population or the specific patient pool, collaborate with hospitals, and move ahead.
I guess Madhavrani madam has got her answer. Sir, with your permission, I would like to ask two more questions from participant side. What host system are you using for the transaction and expression of cancer molecules?
So initially, in general, not only cancer, a large case of the different kind of biopharma mAbs, generally CHO cell line, Chinese hamster ovarial cell line are chosen. Classically, traditional platform is used for different kind of IgG therapeutic drugs at the stage of as a host cell to start with.
Okay, sir. Sir, the same person is a question is manufacturing license is different from market authorization. It's from Rupak Kumar, sir.
See, market authorization is actually a large. It's quite. See, market authorization means you have already got the manufacturing license to make it. That's how we are making it. But market authorization is for making it at the commercial scale. So, at manufacturing license, you will have to have it well before. So, if you look at the presentation workflow, we have a preclinical. We had a discovery, early stage, and preclinical, and then you have the clinical trials material you will have to prepare in your facility. Then only you can do clinical trials, right? So at this stage of preclinical, you take tox batches. They are also batches being manufactured, toxicity batches, and post that you in the manufacturing facility you make your manufacturing batches at the clinical trial material one. So now with your early safety and efficacy data from the R&D data package, which we are going, if it's for a new drug, for an example, investigational new drug application where you're submitting, that's where you actually get further license for manufacturing it at the clinical trials. That's quite early. And market authorization means you already have everything. So you would be making at the commercial scale. It is not on the clinical scale. Clinical scale is like you'll be making a clinical trial 1 material, trial 2 material. Trial 3 material is even larger than that. But when you have MA, that means you are making at the commercial scale. So that is a huge fermenter capacity you are making it at your facility. So that's how they are distinguished.
Thank you so much, sir. I guess all questions till now posted in the chat box has answered very from the expectation side of the participants. And once again, thank you, sir, for the insights you have given in this particular platform. Still, it is a vast knowledge. We need more and more like studies and these kind of sessions from your and these kind of lecturers and resource person should be more so to enlighten us in these areas. Dr. Devashish, sir, can I hand over you the last session?
Yes, absolutely. Thank you, madam. And once again, we are deeply grateful to both these speakers today for sharing some great insights about the industrial aspects of biopharma and drug discovery. Both are very eminent persons with vast experience now in the biopharmaceuticals, research, biosimilars, okay, and so on. So, today's session, we had, as said, two industrial experts, where Dr. Swati initially focused mainly on the biopharma discovery development, and she has also very nicely represented or shown her new startup or venture, which is focused on bioconsultancy services and the different aspects of the entire biopharma drug development or discovery process that they are able to handle. So it's eye opener for many I think, new entrants in the industry. She has also shown elaborately on some of the very innovative, the research or the therapeutics research that they are into now. Next, Dr. Avijit has again given a very nice overview of the biopharma development. He has shown explicitly the challenges that are faced in the biopharma drug discovery and development, and especially in that regard, how automation can fill the gap, okay, in terms of saving time, resources, okay, and of course, at the same time, highly upgrading the quality aspects of the research, the process, and finally the products. So once again, we are deeply grateful to both these speakers today, these industrial experts today. And again, I have been associated with them for quite some time. So, it's really good, to have their experience, and participants also, I think, can now.
Can we have a group photo, sir? Just 2 minutes.
Yeah, so let us have that, and let me conclude that, okay, with the session, and we can have these group photos in between. So um, so the participants or also can actually look into the transition, okay, from the initial part of our this FDP program, say for first 3-4 days, where it was focused mainly on the basic research. Now it's more moving towards the applied and the industrial aspects. So that's how research is all about, right? So if you are not able to come up with some kind of products with the research, it's of no value. So that's how we have actually tried to design it, from the basic to the applied part of the the research or the drug discovery. And we are really thankful to all the participants also who have taken time to associated with us very diligently till now. And even today, where it's a holiday in many parts of India, and such an important festival to be associated with us till this late night. So, without you, I think nothing actually is of value. The sessions, the discussion, nothing. So, you are the I can say the main uh, building blocks or the pillars of this entire FGP program. So we heartily, I and my coordinator, are really grateful to each and one of you for joining and making these interactions really useful. So with these words, I just like to remind you once again of the feedback link, which will be open till tomorrow 2 p.m. So kindly fill that link, and of course the test about which we have described earlier in the meeting. Please note that the test, as again said earlier, is important to get your certificates, and it will be directly it will directly come from AICTE, okay, based on your the attendance and the your test response. So kindly request to all of you again to please attend that test, which is very important tomorrow. If there is anything further clarification, we'll again pass on to you successively in the chats and in other way. So thank you all once again, and I think, as ma'am told, we can now all join with the resource person for a group photo. So which is a nice way to end Sankranti today.
Thanks, Dr. Devasis, for this nice closing notes, and Dr. Moshe Madam for this summary of the discussion. So thanks, everyone, and happy Makar Sankranti to everyone.
Yeah, so I just request all the participants who are here in this meeting platform now to kindly switch on their videos so that we can again say goodbye with a very happy, nice group picture. Thank you, Vipul sir. Okay, and I request others also, if you're associated and can hear me, please put on your video so that one group photo can be taken. Thank you, madam. Thank you all who have switched on. kindly just for few seconds, please switch on your video, everyone. And if it is really not comfortable, you can just skip. Not an issue. So Parth bhai, you can please take the group photo in the grid view of it. Thank you all again.
Devashish sir, please enlighten the timing. Tomorrow that there is a change in timing in the schedule. It is not 6 o'clock. It is
Yes,
2
yes.
p.m. Kindly also ensure that. And the test timing, that is 6:30. Kindly say that. So
Yeah.
everybody
So
will be enlightened. All those
okay.
schedules are there, but still.
So as as ma'am said, and that's why ladies always have the upper hand. See, that's last thing which I forgot. Ma'am has very nicely said.
No, it's a team. It's always a team. No
Okay,
bias. It's always a
so
team.
no, I'm just acknowledging overall, ladies. Anyway, so as ma'am said, very correctly, please note the shift in the timing. So tomorrow we are joining actually from 2 p.m. So
It is
tomorrow is the
actually the
last.
timing in the schedule. It has been given.
Yes,
So
yes. I'm just saying that. So tomorrow, on the last day of the FDP, that is day 6. Please note the time. We will start from 2 p.m. the sessions. The reason for this that there are after the sessions, there is this time for the for your online test and feedback, which will be from 6:30 to 7 p.m. I'm sorry, 6:30 to 7:30 p.m. 1 hour. So 50 minutes test plus 10 minutes of feedback. Okay, and followed by 7:30 to 8 p.m. validatory session. I request one and all of you, okay, very sincerely, to please join the validatory session. This is a must and very important for everyone who are associated with this FDP program, including the participants, the coordinators, the resource persons, okay, to be present in this valedictory function, okay, so that we can conclude in a nice way. So all of you kindly join that. So I hope you have noted the change in the timings on the last day tomorrow. So if that is fine, we can say finally goodbye and good night today. Thanks for all
Yep,
again
thanks
tomorrow.
everyone. Yep, good night.
thank you all of you. Thank you participants. Thank you coordinator sir. Thank you Abhijit sir. Thank you so much.
Thanks
Thank you
everyone.
all.
Bye.
Thank you. Good
Good
night.
night all of you.
Take care. Parth bhai, you can close the